We observe many types of blood cells rushing by as we attach ourselves to the vessel wall. A scan reveals the many diverse types there are. First, the blood cells are classed into two different lineages, lymphoid or myeloid. The myeloid lineage includes red blood cells, probably the best known of the blood cells. Its function is to deliver oxygen to body cells and take away waste carbon dioxide. It is one of the only cells in the body that does not have a nucleus, which accounts for the indentations on both sides indicating an empty space. The red blood cell discards its nucleus while it is developing to make room for more of the oxygen-carrying protein hemoglobin. Next in the myeloid lineage are a group of related cells known respectively as neutrophils, eosinophils, and basophils. Neutrophils are the better known of the three, and also the most common. They are the first responders to invasions from external threats, such as pathogens. They are extremely mobile and able to squeeze through the walls of a blood vessel to reach harmful invaders. They are able to go through a process called phagocytosis. This means engulfing a pathogen and breaking it down with digestive enzymes. Eosinophils are much less common than neutrophils and combat multicellular parasites by phagocytosis. They also release antihistamines, which counteract the effects of histamine, a chemical that causes an allergic reaction. Basophils release histamine to launch an inflammatory response by the immune system and heparin to decrease the blood's ability to clot. They circulate in the blood. Mast cells are similar to basophils in that they both release histamine to start an inflammatory response. The main difference is that mast cells stay in tissues, while basophils travel through the blood. Both cells are responsible for allergic reactions, where they cause an overreaction to a harmless substance such as pollen. The next type of blood cell is the monocyte. This cell protects against pathogens through phagocytosis. When it exits the circulatory system and anchors in tissue, it transforms into either a macrophage or a dendritic cell, both of which defend against foreign substances through phagocytosis. The last cell in the myeloid lineage is a huge entity called a megakaryocyte. This cell pinches off pieces of its cytoplasm to create platelets, small cell fragments that are essential in order to cause blood to clot. They activate when a blood vessel is ruptured, and they grow arm-like protrusions which help them clump together at the break in the vessel. This is how they cause clots. First in the lymphoid lineage are B cells. They are distinguished from T cells because they mature fully in the bone marrow, while T cells, which we will mention later, finish maturing in the thymus gland. B cells are part of a group of cells called professional antigen-presenting cells. Also in this group are macrophages and dendritic cells. Cells in this group have the ability to phagocytize pathogens and present a peptide chain from them using a protein called Major Histocompatibility Complex II, or MHC2, on their surface. B cells have receptors on their surface that are specific to a certain virus or bacteria and are unable to connect to any other pathogen that it is not specific to. The portion of the receptor that is specific to a certain pathogen is shuffled for each B cell while it is maturing. This way, there are many B cells in our body, each with a different combination to connect to a certain pathogen. There are about 10 billion possible combinations for this receptor, but the combinations that will cause our own body to be attacked are destroyed. Once a B cell finds a match to its receptor, it engulfs the pathogen and displays an antigen, a molecule specific to that pathogen, on its surface using an MHC2. It is now ready to be activated by an already activated helper T cell. There are two types of T cells, helper T cells and killer T cells. Helper T cells control the immune response to a threat. A helper T cell has different receptors on its surface that are similar to B cells, in that they are shuffled during its maturation and are specific to a certain antigen. It is activated by professional antigen-presenting cells, such as dendritic cells. When a dendritic cell senses something foreign, it phagocytizes it and displays an antigen from it on its surface using an MHC2 like a B cell does. Dendritic cells are special because they have the ability to activate naive helper T cells. When a dendritic cell with its antigen meets up with a helper T cell with the receptor for the same antigen, the helper T cell is activated, represented by the glow, and begins to divide into memory and effector helper T cells. Memory B and T cells are just multiple copies of the original cell that last for years instead of days like normal cells. The purpose for these cells is to cause the immune reaction to start sooner if the pathogen invades again, because there will be a greater chance of them bumping into a B or T cell specific to them. Vaccines use this ingenious system by introducing a harmless form of the pathogen to cause the body to create memory cells for that specific pathogen. This will give the immune system a much better chance at defending against the full-strength version of the pathogen later. Effector T cells activate B cells that are displaying this specific antigen, and they produce chemicals, shown in the turquoise particles, which cause activated B cells to work harder at their immune response. Activated B cells divide into B memory cells and B effector cells, also known as plasma cells. Plasma cells start producing about 2,000 antibodies a second. These antibodies are specific to the original pathogen and will bind to that pathogen if it hasn't invaded the cell yet. This basically marks them for destruction from cells such as macrophages. Killer T cells, on the other hand, destroy pathogens that have invaded cells, such as viruses. They bind to MHC1, which all cells have. Only professional APCs have MHC2. MHC1 displays proteins from the cell. If the proteins are abnormal, as in the case of an infected or a cancerous cell, killer T cells with the exact combination for that protein bind to the cell. When an activated effector helper T cell starts producing chemicals, it activates the killer T cell, which kills the infected cell by boring holes in its cell membrane and injecting it with cytotoxins to kill it. It then divides into effector killer T cells and memory killer T cells. Effector killer T cells repeat the process of destroying cells infected with the original virus. This lengthy explanation shows just how detailed and complex the cells of our blood are.